In this lecture, we will discuss histology, specifically how to identify tissues. A few helpful hints for the exam. You will need to be able to identify both general and specific tissue types. There are only 4 general tissue types: epithelial, connective, muscle, and nervous tissue. However, there are many specific tissue types. So if you look at your lab list, you've got simple squamous, epithelial tissue, transitional epithelial tissue. You've got fibrocartilage, connective tissue. You've got, my mind went blank. You've got lots of them. Anytime you are asked for a general type of tissue, you only have 4 answer choices: epithelial, connective, muscle, nervous tissue. Anytime you're asked for a specific type of tissue, that's when the rest of those names come into play. So make sure you're keeping those straight. Also, you're going to want to make sure you fully name a tissue. So you have loose adipose connective tissue. That is the full name, right? You've got your connective tissue, which is the general type. Your loose adipose is the specific type of tissue. Simply writing adipose or adipose tissue will not get you full credit. So just keep those things in mind. Now, you're also going to want to know locations and functions for the tissues on your lab list. I would encourage you to perhaps make flashcards with a picture of the tissue on the front and then the locations and functions on the back so you can use them to study. And then don't forget about the additional structures or cells associated with some of the specific types of tissues in your lab list. So any tissue that has an extra bullet point within the little table, that's an additional structure or cell that you're going to need to be familiar with. So for example, blood tissue, you don't have to just know that it's blood tissue, but you need to know the difference between a red cell, a white cell, and a platelet. Alright, so epithelial tissue. We have 4 main functions: protection, absorption, filtration, and secretion. So for protection, we're really talking, generally speaking, we're talking more about stratified tissue, stratified squamous epithelial tissue. You find this anywhere that is exposed to a lot of repetitive motions or abrasions, anything like that. So this is the skin itself, okay, as well as the linings of the mouth and the vagina. Absorption, filtration, and secretion, we tend to see those functions associated with the simple epithelial tissues, the single layers, as it's easier to move substances across one layer of tissue versus many layers of tissue. Now, I hinted at this. When we name epithelial tissues, we have 2 things to keep in mind: the number of layers within the tissue and the shape of the cells that compose the tissue. So we only have 2 options for number of layers, because you could have 1 layer or you could have more than 1 layer. If you only have 1 layer, you are in simple epithelial tissue. If you have more than 1 layer, then you are a stratified epithelial tissue. Now, if you do have more than 1 layer, if you are a stratified tissue, when we name you by the shape of your cells, we always look at the apical surface, the top layer. We don't look at the basement membrane or the bottom layer, and I'll show you why here in just a second. So let's look at our shapes real quick. We've got squamous, which that word really just kind of means nothing to us, but they're really flattened. I think they look like little fried eggs. Okay. Then we have cuboidal and columnar, and fortunately for us, these really do look like little cubes or little boxes, and then columns respectively. So these 2 are pretty well named. Now, we mentioned that we're not going to name these at the basement membrane for stratified squamous, and I said I'd come back to why. Here's why. If we look at this image specifically, You can tell very quickly that we do have multiple layers, so this is a stratified tissue. But if we looked here at the bottom, if we looked at the basement membrane, we could be convinced that this would be stratified cuboidal epithelial tissue. These cells at the very bottom layer, the basement membrane layer, they almost look like little cubes. Okay. However, if we look at the top, we will see very quickly that no, those aren't cubes. These are those squamous cells, those little flattened cells. So why do the ones at the bottom look different? Well, it's pressure. All of these other layers are pushing down on this bottom layer, and it kind of gives them a different shape for appearance sake. So again, anytime we're looking at stratified tissue, we're always looking at that top layer when we determine the shape of the cell. And you will notice for all of these, we are mixing and matching. You have to pick both words, simple squamous or stratified squamous, simple cuboidal, stratified cuboidal, simple columnar, stratified columnar, or even pseudo-stratified columnar. Okay, so pseudo-stratified columnar. Pseudo means false, so false stratified, so fake multi-layer. Okay, so if you look at this very quickly, okay, the nuclei are all over the place. You could trick yourself into saying, oh, that's multiple layers. Okay. However, if you look at all of these cells individually, everybody touches that basement membrane. Okay. Even this skinny little cell is still touching the basement membrane. So this is technically still just one layer of cells. Okay. So one at a time. Simple squamous epithelial tissue. So simple squamous tells us that we have one layer of flattened cells. This is an image of lung tissue. This is just a classic example of simple squamous epithelial tissue. You will notice that we do not have layer upon layer upon layer. And it might be a little hard to see, but all of these tiny little dots, each of these little dots is a nucleus, so it is connected with an individual little cell, and they are just spread out one after another, okay, making this particular configuration. You will notice all of this empty space, okay, all of this empty space, it really just is empty space, okay. This is, like I said, lung tissue, and this is where all of the air would be coming in from the outside of your body into your lungs, and it would be going through these epithelial tissues, and it would be getting into your blood. Now, you've got your locations and your functions. Please make sure that you are really only studying locations and functions within your lab list. Okay, so these notes, haven't really changed these notes in a while, but we could have updated your lab list. So there may be additional locations and functions on here that we've just taken out of your lab list. It's not that any of these are wrong, okay? We've just tried to help you narrow down some of the more particular locations and the more important functions. So just make sure you're going by what's on your lab list. All right, simple cuboidal, still have one layer, but now we have those boxes, those cubes. And if you look at this too quickly, you say, oh, these aren't cubes, these are circles. Okay. Well, yes, they are circles, but each of these giant circles is a kidney duct, okay? And then each of these kidney ducts individually is made of cuboidal cells, okay? So because they're arranged in a circle, they aren't perfect boxes anymore, okay? But they are not columns, definitely, and they are not flat like a squamous cell. So a lot of times when you are identifying epithelial tissues, it's really a process of elimination. Is it flat? No, then it's not a squamous cell. Is it a column? No, then it's not a columnar cell. Well, what's left? Oh, well, it must be a cuboidal cell. Now, columnar cells, simple columnar specifically, you will notice that these nuclei make one fairly nice symmetrical line. Okay. That is a giveaway for simple columnar epithelial tissue, and I'll show you why on the next slide. But notice how nice and symmetrical these nuclei are. This particular tissue also has cilia. Okay, these cilia are for moving other substances. Okay, Now, I mentioned the nuclei being all symmetrical, and I said it'd be apparent on the next slide. So, pseudostratified, columnar, ciliated, epithelial tissue. Whew. Okay. Now, first of all, notice on these nuclei. Okay, they are all over the place. They're all over the place. Okay, so that is your first giveaway that this is not simple columnar epithelial tissue. Okay, so one nice line for simple columnar, not nice line for pseudostratified columnar. Okay, now that is honestly your biggest hint because, and these are a little harder to see, but this tissue also has cilia, just like our previous slide, so that's not really a distinguishing characteristic. And then we have goblet cells here, these big white cells. Goblet cells are good at producing and secreting mucus. Okay, now we didn't see any goblet cells in this example of simple columnar epithelial tissue, but if you get your tissue sample from your digestive tract, for example, you would definitely also see goblet cells there as well. And so just because this example does not show goblet cells doesn't mean that they don't exist in simple columnar. And so goblet cells actually aren't a distinguishing characteristic between pseudostratified and just regular simple columnar epithelial tissues. Okay, you're really looking at the nuclei. All right, stratified squamous epithelial tissue. This comes in two varieties. You've got non-keratinized and you've got keratinized epithelial tissue. So non-keratinized epithelial tissue, stratified squamous epithelial tissue. This is, as we've mentioned, one of the tissues that you're going to have to look at the apical surface to determine the shape of the cell. And so if you look way up high, you can see that these are very flat. However, if you were looking way down low, they don't look nearly as flat. In fact, they almost look round. Okay, and that's not what we're looking for. We're looking for the top. Okay. Now, the non-keratinized versus the keratinized. Okay, if we compare these two pictures, you'll notice that we have an extra layer over here. That's the keratin layer. Okay, this layer of keratin is good for waterproofing. Okay, so anywhere that we don't want the tissue to dry out. Okay, so keratinized, you would find keratinized stratified squamous on your skin. Okay, non-keratinized, we are less concerned about drying out because it's on the inside of our body. So you would find this in your esophagus, your oral cavity, even the vagina. Remember, the purpose of the stratified squamous, these multiple layers, is to act as protection. Okay. All right, transitional epithelial tissue. Now this one's a little different. We don't say simple transitional, we don't say stratified transitional, we just call it transitional epithelial tissue. This is also a little different because these do not have the typical appearance of the squamous, the cuboidal, or the columnar cells. I think these look like little balloons. They're very, very round. And this may or may not be a little difficult to see, but if you look at some of these other ones down here, they're just very round. Okay. If you look farther back, okay, and I try to highlight it here. If you look farther back in here, this is not transitional tissue. Okay, way back here. So again, we're looking for an apical surface for epithelial tissue, and the apical surface is always going to have some sort of empty space up at the top, and so the top this time just happens to be kind of this star-shaped appearance, and that's okay, but we're looking for the shape way up here. Okay, and so you've got those nice round little balloons. These line your urinary tract, your ureters, your bladder, the renal pelvis of your kidneys. It's really good for stretching, so these little balloons will just kind of stretch out flat when your bladder's full, and then when your bladder's empty, they kind of shrink back up and they look like these little balloons again. All right, the layers of your skin. So you have three layers to your skin: epidermis, dermis, and hypodermis. Your epidermis is that stratified squamous epithelial tissue that we saw just a couple slides ago. Okay, all this purple layer way at the top would be the keratinization, and then you've got the pink stratified layer as well. Okay, so you'll notice that this is all included in your epidermis. Then we come to your dermis, the next layer. This is dense irregular connective tissue, and then your hypodermis is adipose connective tissue. And so we'll hit connective tissue here in just a minute, but you are going to want to be familiar with what these different layers look like on images and slides.